Welcome to New York Public Health Now, where we talk about the why so you can decide what to do. Hello, I am Dr. Jim McDonald, Commissioner of the New York State Department of Health. So welcome to the 14th floor here in Corning Tower in Albany, and I'm joined today by the Acting Executive Deputy Commissioner, Joanne Morne. Joanne, how are you today?
I'm doing well, thank you.
Yeah, and good to have with us as a guest Dr. Paul Masters of the Wadsworth Laboratory here at the New York State Department. So good to see you, Dr. Masters. How are you today?
Thanks for having me on, Commissioner McDonald.
Yeah, and great to have you as well. You know, it's interesting. I've read about your career, which is quite impressive. I would almost dare say legendary. It's quite frankly humbling to have intellects as great as you here at the department. Just really want to say that out loud here, which I think is great. So let's get started.
All right. Well, I think with that introduction, why don't we start with tell us about yourself.
Okay, well, and thank you for that very generous introduction. I'm a proud graduate of Yonkers High School. From there, I went to Cornell University, and I was a chemistry major. And I discovered toward my senior year that I was most interested in the chemistry of living things. Fascinated by how proteins and nucleic acids assemble themselves into these long polymers. So I then went on to Brandeis University, where I got a PhD in biochemistry. And again, my focus sort of drifted a bit to an interest in molecular biology and viruses. I did 2 postdoctoral stints where I could combine those 2 interests, one at the University of California at Santa Barbara, and the other at the Roche Institute of Molecular Biology, which was in New Jersey. And then finally, I joined the Wadsworth Center in 1988 to perform basic research in the molecular biology of coronaviruses, which I've done ever since, supported by grants from the National Institutes of Health.
You're Wadsworth's in-house coronavirus expert, and it's just interesting. I think, I've been a pediatrician over 33 years, and, when I treated children with a cold, I didn't usually find out if they had a coronavirus. It didn't really clinically make that big a difference, but boy, that changed after the pandemic and underway. But I'd kind of like to back it up a little bit and just start with, what is a coronavirus?
Well, coronaviruses are a family of viruses causing respiratory and gastrointestinal diseases in humans and also other animals, and additionally in birds. Among the animals having coronaviruses includes both wild and domestic animals. Coronaviruses have been on the radar of veterinarians for ages because they cause a lot of diseases of agricultural importance. But we're most concerned with humans, of course, and there are 7 human coronaviruses. 4 of these are so-called endemic coronaviruses. They've been among us for at least a century, probably more in some cases, and generally cause common colds. Some of them, sometimes that can be serious, go on to pneumonia. One of them causes childhood croup, but generally not life-threatening. However, there are 3 other coronaviruses that have emerged in the past 2 decades, and these cause very serious disease. The first of these, some folks may remember, in 2002, 2003, this virus that came to be called SARS coronavirus 1, severe acute respiratory syndrome coronavirus 1, caused an epidemic that fortunately the virus was eradicated by the enforcement of strict public health measures. And so that virus is no longer among us, and that's a tremendous victory. About a decade after that, in 2012, another deadly coronavirus emerged, and that's called Middle East Respiratory Syndrome coronavirus. And it's a virus that is pretty common in camels and jumps to humans sporadically and causes a very severe pneumonia with something like a 30% case fatality rate. And for reasons we don't understand, this epidemic is confined to the Saudi Arabian Peninsula and has really not moved elsewhere, fortunately. And then finally, in another 10-year gap or so, in 2019, SARS coronavirus 2, the virus that causes COVID-19, emerged, and everybody's familiar with that story.
Yeah, absolutely. The last 3 years have been exceptional, right? But you also have been with the department, as you said, since 1988. Can you talk to us about the work that you were doing before COVID?
Sure. My focus has always been the molecular biology of coronaviruses, and particularly studying a model coronavirus mouse hepatitis virus, which only infects mice and fortunately not us. And so my focus has been on what are these viruses made of? What are all the molecular details of how they infect cells? I'll tell you a bit more about that. I first have to tell you more about coronaviruses. These are the virions of coronaviruses, that is, the viral particles, are roughly spherical. They're surrounded by a membrane that they steal from the host cell, and they're about 100 nanometers in diameter. What does that mean? How small is that? If you lined up 250,000 of them in a row, that would equal about 1 inch. So that's how tiny these things are. The virions, the viral particles, have a relatively small number of proteins, and the most prominent among these is a protein called the spike protein that projects from the surface. Multiple copies of the spike protein project from the surface of the virion and give it, to some people's minds, the appearance of a corona, and hence the name of the family. But the business of the spike protein is to bind to particular receptors on the cells of hosts and to initiate infection. So the spike protein binds the receptor, undergoes these contortions that allow the guts of the virion to end up inside the cell and initiate infection. And another important protein in the virion is something called the nucleocapsid protein, and that wraps up the genetic material of the virus, the genome of the virus. And unlike us and all living things we encounter, the coronavirus genomes and most viruses have genomes made up of ribonucleic acid, RNA, not DNA. So we're all DNA-based organisms, and so is every living thing you've run into. But most viruses use RNA as their genetic material, and this gives them particular vulnerabilities, but also particular advantages. RNA is quite different than DNA. You're familiar with the story of the double helix. RNA can fold up into a whole variety of three-dimensional structures that can be recognized in different ways by different proteins. So not only the sequence of RNA is important because it codes for proteins, but also the structures are important. And so what we did relatively early on was we developed the first reverse genetic system for coronaviruses. That is a system by which we could plant specific mutations into the genetic material of the coronavirus and use this to study basic principles of protein function. How do all these proteins get together to assemble a virus? And what roles do other proteins that don't get assembled into the virion play during the course of infection in cells? And we also use this genetic system to study RNA sequences and structures in the viral genetic material and how proteins interact with those to replicate the RNA, make new copies of RNA, and ultimately recognize the RNA as the thing that needs to be incorporated into virions. So we just want to learn fundamental mechanisms of the viral life cycle so that we can potentially identify molecular targets, weak points against which therapeutic agents could be targeted.
Yeah, a wonderful review of the basic science of coronaviruses, by the way. That was great. And but I think it's important to understand you don't just do basic science. You do science that leads to actually things we use. And I want to go back in time to 2020, in the beginning of the pandemic in New York State. And, you have a lot of extensive knowledge about coronavirus research. And yet that had a lot to do when we were jumpstarting New York's COVID response efforts in those critical early days of public health. And I know it wasn't just you, but can you talk a little bit about how you and the team at Wadsworth helped when it came to, the clinical sampling and the antibody testing? How did that come together?
Yeah, the critical thing in the earliest stages of a disease outbreak is to have a test to determine, first of all, who is infected.
Yeah, what are you dealing with, right? Like, you got to have a test, right?
That's the diagnostic capability. Right. And secondly, to determine how far the disease has spread. Right. And so that's the surveillance capability. And so in late February 2020, before it was clear that COVID-19 had even arrived here, I was able to advise members of the Laboratory of Viral Disease in the Wadsworth Center, which is directed by Dr. Kirsten St. George, and we worked on what are the best sequences to target by PCR, polymerase chain reaction, in this test to be developed, and how to ensure that it was specific for SARS coronavirus 2 and not for all the other human coronaviruses that are floating around.
Right.
And so that's critical. And on this, I work with two terrific young scientists in the Laboratory of Viral Disease, Sarah Griesmer and Darrell Lampson, and they're the ones who, after we did all the groundwork, they're the ones who went on to establish the test in the laboratory in a real-time, fast turnaround platform that was capable of very high throughput. And as importantly, they validated the test. They went through the whole validation process to ensure that it was accurate and sensitive and specific. And as you know, the Wadsworth Center was the first state public health lab in the United States to have an FDA-authorized test that New York State could use for diagnosis of COVID-19. And this was very important because in the United States, New York City was the epicenter of the outbreak in the early stages.
And I think that's an important point just to drive home is the coronavirus test. Wadsworth was really the first place to not just develop it, but actually utilize it. I mean, that's translating science into action. And it's funny, you talked about you and your colleagues' work, but it sounded a lot like Star Trek medicine, to be really candid with you. It really sounded very much like type of stuff you'd see on Star Trek, because it's just, I'm an old doctor. I know I don't look old. Thank you very much. But and when I was in medical school, I never saw this type of technology being developed. And this is part of the fun thing about leading the Department of Health in New York is we have Wadsworth Lab, which is people like you and your team, which Humphrey just talked about. This is the test that changed the pandemic for the United States. I don't think people appreciate the significance of this.
I think it was certainly a great accomplishment early on, and to see the way the team worked. I mean, all the members of the Laboratory of Viral Disease, plus volunteers from all over the Wadsworth Center, worked around the clock for many months in all aspects of the testing, going from sample accessioning of patient samples to RNA extraction to performing the actual PCR test, and then, very importantly, to reviewing and reporting the results out, and then answering questions from the public and from healthcare providers and from elsewhere in the department. So it was a tremendous group effort, and the number of people involved was just amazing.
There's so much to be proud of based on the work that was done, and certainly led the way, not only in our state but across the nation. And as you were talking, can you just give us a little bit more? I know for myself, I had the opportunity to do work as it related to antibodies, during our response. But the distinction between the antibody testing and the PCR testing.
Oh, certainly. Yeah, I neglected to go into antibody testing on the previous question. So PCR looks for the viral nucleic acid, the viral RNA, and provides a current infection diagnosis. If you're PCR positive, you are infected. But antibody testing gives a broader view. It tells who has been infected both in the past and in the present. What has your immune system seen? And it may also, in the early days, it could also be used to distinguish between individuals who had been vaccinated as opposed to individuals who had been infected. And so I was able to help at the start in setting up antibody testing because I had, buried in my freezers, purified nucleocapsid protein from the original SARS coronavirus 1. And it turns out that cross-reacts very nicely with antibodies to SARS coronavirus 2. So that got things started initially in the diagnostic immunology lab. And I and Dr. Lily Kuo in the arbovirus laboratory immediately went into cloning and production of SARS coronavirus 2 proteins in bacteria and other cell types. So we could produce gobs of these proteins for the antibody tests. I did the easy one. I did the nucleocapsid protein, and Lily Kuo did the much more complicated spike protein, the S protein. Both of these were then purified by Dr. Karen Chave in the Wadsworth Protein Purification Core, and then sent on to the Diagnostic Immunology Lab for assay development. And they developed a whole range of assays using this microsphere multiplex technology, which allows you to look at multiple antigens and incorporate multiple controls in the test at the same time simultaneously.
I mean, I find this all fascinating. It's so interesting. And I mean, part of me wonders, why'd you keep that original coronavirus purified protein in your freezers. I mean, you've been here a long time, since 1988, and I'm so thankful you kept it in your freezer, but I think it's a good example of like, really good thing that you kept that around. I mean, how did you know we'd need it someday?
Well, we didn't.
Yeah, it's just good fortune, I guess, right?
Yeah, and when you go to the trouble of making such a valuable reagent, you keep it as long as you can. So,
I think that just speaks to a little bit about just the perseverance of science. And it's funny, I don't know that in science we get as lucky as much as people think we do. I think it's about forethought, perseverance, and planning that puts us in a place where that actually happens. Sometimes I say around that probably chance favors the prepared, and you impress me as someone who were prepared. So I think that's to the credit of you, but also benefit of the larger population. Speaking of the larger population, it got me thinking about antibody testing in general, and it just made me wonder, like, how does antibody testing, how could it help us inform public health decisions at the population level?
Antibody testing is the determination of what viral protein or proteins a person's immune system has developed antibodies against. And so, most important, it measures the extent of infection, as I said before, past and present in the population. And more generally, it can give us information about the degree of immunity in the population. But with coronaviruses, this is a little less clear, and it's not known how long that immunity lasts. So these are things that are still being
That's really the big question,
worked out
isn't
on
it?
the run. Yeah,
right. I mean, it's called a common cold for a reason. But that's one of the things about immunity is like, you get long-lasting immunity from some illnesses that'll last you forever. For coronaviruses, we've never really thought that was the case, and so that's part of why people get colds every year, it just is what it is.
It would be very nice to have a second-generation vaccine that protected us against all variants of SARS-CoV-2 and protected us against all the common coronaviruses. And there are people working on that sort of thing. So just as they're working on a vaccine that would cover all strains of influenza virus so that we wouldn't have to be boosted as frequently, perhaps. But to get back to antibodies, I really need to give credit to Monica Parker and Linda Steyer in the Wadsworth Center Bloodborne Viral Disease Lab. They developed an assay using finger-stick collected dried blood spots. So you could get a greater participation in the population if people knew they just had to get a finger stick and collect a few drops of blood on a piece of filter paper as opposed to having their blood drawn. And so Monica and Linda developed a high-throughput multiple antigens multiplex assay using this, and they used that assay to conduct a serosurvey of greater than 57,000 New Yorkers had been surveyed by June of 2020. So this is just 5 months into the pandemic, and the groups they looked at were members of the community as well as healthcare workers all over the state, in most counties of the state. And from this, it was concluded that as early as the end of March 2020, over 2 million New Yorkers had been infected with this virus. And furthermore, since a lot of data was also collected anonymously from the participants, the data could be stratified by geography and ethnicity and age and other factors, and you could use that information to determine how best to serve various groups within the population of New York State. So that's the sort of thing that antibody surveillance can do at the population level.
So there are so many things that it sounds like you had the opportunity to anticipate or to think about in planning. Were there surprises for you as it related to SARS-CoV-2?
There were a whole slew of surprises, some of them too technically detailed to go into. But early on, I think I was as surprised as everybody else by how rapidly this thing spread. I mean, I like probably most of us, were watching what was going on in China on the television and saying, that's not going to come here, right? And wow, it came here, and it came here with a vengeance really quick. Further into the pandemic, the big surprise for me was the rapid scientific progress that was made, especially in mRNA vaccine development. That really was wonderful. And this was made possible by basic science that had been done years earlier. After the emergence of SARS-CoV-2, Jason McClellan at the University of Texas was able to determine the three-dimensional structure of the spike protein in tremendous detail by a process called cryo-electron microscopy. And just as importantly, he was able to tamper with the spike protein and stabilize it in such a way that it would be much more likely to elicit neutralizing antibodies when used in a vaccine. And then the other folks who deserve great credit and have received it are Kathleen Carrico and Drew Weissman, who developed the mRNA vaccine method and who just got the 2023 Nobel Prize in Physiology and Medicine.
I love this basic science history you're giving us, but also just telling us the story behind the scenes of the pandemic. I don't think it's been widely told how many wonderful things have occurred to make what looked normal for us possible, whether it's a vaccine, whether it's a test, and all this to me is just interesting. I'm just curious, I don't know if you expected a coronavirus to be the cause of the next pandemic, and I'm just curious if that surprised you at all. I mean, to say this, like living through H1N1, I think a lot of people are thinking if we're going to have a pandemic based on the 1919 flu pandemic, it was going to be a flu virus or something that would have caused a pandemic. But were you surprised it was a coronavirus that caused this pandemic?
Yeah, frankly, I was. I would have put my money on influenza also, and we're still looking at H5N1 bird flu.
Yeah, we are, aren't we? We pay attention
about,
to that one.
How much does that have to mutate in order to cross over to human populations? And there, people have mapped out pretty well how much influenza virus, for bird influenza, has to change in order to be able to infect humans. But these things do come upon us as a surprise, and it's important to be prepared.
As we think about future, continuing down that road and thinking about the potential for other pandemic threats, how optimistic are you about our continued ability to develop tests or rapidly develop a test?
To rapidly develop a test, I'm extremely optimistic. I mean, to my mind, it's pretty much a certainty. The technology for test development has just proceeded by leaps and bounds, and the range and the ability of testing technologies it just keeps increasing day by day. What I'm more concerned about is our ability to maintain preparedness. We can't afford to get complacent and say, well, the pandemic was here and now it's over, we're all okay. We need an ongoing investment in public health, and part of maintaining preparedness is a need for constant surveillance. And a great example of that is the New York State Wastewater Surveillance Program that's being conducted by Kirsten St. George in the Laboratory of Viral Disease and multiple collaborating laboratories throughout New York State. And they're monitoring SARS-CoV-2 sequences in sewage at a depth at which they can monitor, what variants are there and what the geographic distribution of different strains of the virus are throughout New York State. And also, they can be, because of high throughput sequencing, they're able to look at the whether a new previously unknown variant is emerging. And I think the same technology can be expanded to many other viruses, and with great benefit to us.
As we get ready to close out our episode, and one of the things I think about with this whole idea of a podcast for us was talk about the why, so people can help decide what to do. But I think one of the things I'm taking away from our conversation is when you talk about the why of just testing in general, I hope that our listeners can understand there's a great deal of molecular biology and sound science that makes our testing very purposeful, very reliable, and very much something people can trust. And I think that just speaks to just understanding just the power of science and the power of just, quite frankly, continuing to ask the questions, be prepared about, why you can trust the testing that we do here at the Wadsworth Laboratory and the impact it has throughout the United States. So as we get ready to close, Dr. Masters, it's been great having our conversation with you. Do you have any final thoughts for us today?
Well, yeah, I'd like to say that I've been very lucky to have spend my career at the Wadsworth Center. I've been able to work alongside colleagues performing cutting-edge research in the biomedical sciences and in the environmental health sciences. And at the same time, I was working alongside outstanding public health scientists who are developing and employing state-of-the-art methods to address the major health challenges of our time. And this is a unique model for a state public health lab. You don't see this anywhere else in the country. This integration of diagnostics and research, this model was framed by Augustus Wadsworth, who was the first director of the lab way back in 1914, over a century ago. And I think it will continue to serve New York State very well into the future.
Thank you, Dr. Masters. So, Joanne, I learned a lot today. How about you?
Oh, always. Absolutely, Dr. Masters. It's been a pleasure, and thank you so much for sharing your knowledge and your history.
Well, thank you. I really enjoyed it.
Yeah, Dr. Masters, thank you so much for joining us today. It was great to have you here. As we close out our episode, we would love to hear from you, our listener. If you have an idea for topics we should talk about on our podcast, let us know by email. publichealthnowpodcast@health.ny.gov. Let me repeat the email address, publichealthnowpodcast@health.ny.gov. Keep an eye out for the latest New York Public Health Now episode on your favorite podcast player like Apple Podcasts, Overcast, Spotify, YouTube, and Google Podcasts. Tap the subscribe button or follow button to be notified when a new episode drops. And to easily find our podcast, search by our podcast title, New York Public Health Now, or keyword NYSDOH. Thank you very much. I hope you have a good rest of your day.